Okay, so hello everyone. Welcome to the second week of the special lecture series. My name is Min Zhang, and I'm a professor of statistics and the associate director of data science at Purdue University Center for Cancer Research. So this seminar series is part of our NCI-funded R25 summer workshop on big data training for cancer research. Today we have three panelists, John Fry, Nadia Lama, and myself. So John has helped with setting up the Zoom meeting and handling all the registrations. So he also records the lecture and posts the recording of our work at our workshop website. So now I would like to introduce our webinar moderator, Dr. Nadia Lama. Dr. Lama is a research assistant professor in the Department of Comparative Pathobiology at Purdue. She has been the manager of the Purdue site of Collaborative Core for Cancer Bioinformatics since 2015. Dr. Lama has a background in biochemistry, molecular biology, and computational biology, and she actually got her PhD in 2015 from Purdue. So her research interests focusing on single-cell data analysis and data integration with special interest in immunological diseases of the prostate. So with that, I will hand it over to Nadia.
Thanks, Min. Welcome, everyone. So for the duration of this seminar, we'll keep our mics off and muted. And if anyone has a question at any time during the seminar, please use the Q&A function, which is located at the bottom of your Zoom screen. And also, if you see that someone else has asked a question that you like or would like to have answered, feel free to upvote that question using the same Q&A function. And those questions that are upvoted will sort of bubble to the top of the list and have a better chance of being answered. And so now I'll hand it back to Min.
Thanks, Nadia. It's my great pleasure to introduce Dr. Warren Kibbe. So Dr. Kibbe is Vice Chair and Professor of Biostatistics and Bioinformatics at Duke University. So his research interests includes data representation for clinical trials, especially improving computability and interpretability of biomarker and eligibility criteria and data interoperability between medical records and decision support algorithms. So Dr. Kibbe has been proponent for open science and open data in biomedical research and helped define the data sharing policy for the NCI Cancer Moonshot program. So before joining Duke, so he served as an acting deputy director of the NCI and was the director of the NCI Center for Biomedical Informatics and Information. He was one of the architects of the NCI Genomic Data Commons Initiative and a co-founder of the Cancer Informatics for Cancer Center Society. So Dr. Kibbe is an MPI on the NIH-NIMHD funded REDX Underserved Population Coordination and Data Collection Center, and also an MPI on the NIH-NHGRI funded Duke Center for Combinatorial Gene Regulation. Today, Dr. Kibbe will speak on something related to our current disruptive life, COVID-19 and cancer. So please join me in welcoming Dr. Kibbe.
Great, thank you. Thank you, Min. Thank you, Nadia. So I want to talk a little bit, not about either of those two projects that Min just mentioned, either the RADX UP initiative or what I've been doing with with NHGRI and understanding the non-coding genome. What I want to talk about instead is something I've been involved with for the last bit more than a year at this point, and that's the National COVID Cohort Collaborative. And it's gotten sufficiently mature that now there are a lot of different disease areas and disease groups that are using N3C as a resource, a big data resource. I'll talk quite a bit about what N3C really is. But then I'm going to talk a little bit about one of the projects that's looking at oncology in particular, and using the N3C as a way to understand outcomes for cancer patients. So let me just tell you a little. I like to go through the objectives. So I'd like to go through and define really what real world data means, because N3C is a real data resource. Some of the drivers behind open science. Take you through the N3C data enclave and how it's really been assembled, how it's been harmonized, what kinds of questions it's good at answering. A little bit about data access and security, and I may skip over some of that in the interest of time, and then oncology research in N3C. So I want to thank a number of people who've been part of N3C for giving me lots of slides. Chris Shute and Melissa Handel are the PIs for N3C. Umit Tapalagu at Wake Forest, and Noah Sheriff-Feldon at UAB, so University of Alabama at Birmingham. It's their work I'm going to present, looking at cancer outcomes. And then Frank Rockhold at Duke, he's a big open science proponent, and I'm going to talk a little bit about some of the things that he's been doing around making clinical trial and patient data more accessible. So the take-homes I want you to have for N3C and oncology is that N3C is really a unique resource in the country for understanding the effects of COVID-19 on cancer outcomes, and that it really is the largest COVID-19 and cancer cohort in the US, and it's certainly the largest COVID-19 resource. So I'll come back to that. I'll make those points toward the end. So something I want to do is just define for everyone what real world data is. I think this is one particular definition. It's actually very much in alignment with what the 21st Century Cures Act that was passed back in 2016, how it was written. And it's really all the data that's collected around a patient in the performance of their normal health care. And that could be prevention visits, it could be an annual exam, or it can be involved in providing some kind of treatment. So and you can see a sense here of all the different sources of data, including things that may be posted in social media, digital health, and then the more classic EHR kinds of data. So that's that's when I talk about real world data, this is what I mean. And I think just to emphasize that current sources of data that are found in the EHR include things like molecular data. So sequencing, could be proteomics, could be a lot of different things, pathology data, imaging data, lab data, notes, certainly clinical notes, pathology notes, radiology notes, and then a new class, which is really data coming from sensors. And we won't have much, I won't really talk about sensors again, but again, it's an incredibly important way of understanding health. So I think as we're doing all that, we've come to realize that AI is artificial intelligence and machine learning really is changing our ability to both harness all that data and make sense of it. There's been a lot of buzzwords like trustworthy AI. What does that mean? How do we track provenance of data so we know where it's been, who's acted on it, and what it's good for? And there's a lot of discussions, and I'll come into this from an open science perspective around reusability and reproducibility. And those two things are, at least in my mind, quite different. They're related, but they're different. I think it's also really important, and the last year has really reinforced this, that as we start using digital health, we start looking at precision medicine, we use real world data, that these things really can both transform healthcare, but we have to really pay attention to the limitations of what we're doing and make sure that we're always using a health equity lens to interpret that. Because it's so easy for us to go down the wrong path and actually reinforce inequities in the health system rather than make the health system really work for everyone. And there's this cute little graphic at the bottom. I can't, of course, I can't take credit for that, but there's a big difference between equality, where we're really lifting up everyone to a certain level, and equity, where in fact we have the same impact on different individuals. So it's a cute way to distinguish between equality and equity. And what we've come to learn is that equality isn't really the goal, it should be equity. I won't really talk too much about 21st Century Cures Act. I was lucky enough to be asked to give a presentation last year, and I talked a bit about the cancer moonshot and precision medicine activities that were part of the 21st Century Cures Act. I want to mention a different side of the 21st Century Cures Act, though, and that was really a driver for the FDA to get more involved in using real-world data and real-world evidence to understand outcomes in new therapies, and also provide the opportunity for doing new kinds of therapeutic designs in drug development, as well as that outcomes piece. So just to be aware that there's a lot of push behind real world data now. But as I mentioned at the beginning, it's not just about real world data. It's also about things like open science, data transparency, data access, and those words I used earlier, reusability and reproducibility. So there's been a long discussion. It predates actually any of the activities that I highlight here, which is how do we make research data, research findings, and how do we disseminate those findings in ways that really encourage innovation and allow us to do better in healthcare. So here's a few of the projects that are focused on doing that. The Open Science Project, the FAIR principles, which were released. Boy, I think it goes back to 2011, but the publication I quote there is from 2016, and then more recently the trust principles. So FAIR is findability, accessibility, interoperability, reusability. And then trust kind of goes along with that, and that's being transparent, showing responsibility, having a user focus, thinking about sustainability. That's really hard for data sets. And then the appropriate use of technology. And both of those are Nature articles. So some of the challenges thinking about open science, particularly in the context of patient data, and remember now, I'm going to talk a lot about N3C, and N3C is a resource of patient data, is we need to do this in a way that balances patient privacy. We want to, of course, make sure that we maximize academic credit. We want to preserve intellectual property so that, in fact, the things we discover, we can create things that can actually produce things that are of value to our society. We need to have good data standards. And then, of course, to make all this work, there has to be resources. And exactly how those resources are provided is, it's always a discussion. And then there's a couple principles, right, that researchers and patients alike should gain from open science and from making patient data accessible. Particularly for patients, because their privacy is in some ways at risk. We want to make sure that we're balancing the good that we can provide them with the additional risk of privacy. And then we think it's really important that informatics experts, like all of you in the workshop, and data scientists, like all of you in the workshop, need to be part of the discussion. So I'm going to focus a little bit on clinical trial data sharing. That's not necessarily what N3C is about, but there's a lot of discussion about clinical trial data sharing. And one of the views is that people will sit on clinical trial data for longer than they should. And I think that the GDPR regulations really show that it's incredibly important to recognize that there's an importance of data sharing for patients, for the public, and there has to be a balance between if you will, the credit and the right of researchers with the rights and the responsibility of researchers to the patients. So that's important. And something that we try to do when we make patient level data available, and this does apply to N3C, is we want to make sure that there's reproducibility of clinical research. So if we go back now about a year ago, some of the first papers that were being published on COVID and COVID outcomes, there were a few publications that came out early on that it turns out that the data that they were basing their findings on wasn't what they thought. So that actually created a bit of a crisis in reproducibility. So one of the goals for N3C is to make sure that doesn't happen. We want to honor the contributions of trial participants. For N3C, that's actually not the case. No one is a participant per se, certainly not in a clinical trial. And we want to be able to generate new research findings. And something that's really important is to realize that this is not, these are not new conversations. They've been going on for a very long time. So it is a constant evolution and a move towards making things open. So N3C is, as I've already mentioned, an open science controlled access environment, really for bringing EHR data from a lot of different institutions and making it accessible for research. So it's just a cute graphic of that. And the idea here is we have a number of different institutions covering the entire country, being available for people to do research on it. And I'll talk a little bit about the four common data models listed at the bottom. So the ACT network, the Odyssey network, the CoreNet, and Trinetics, and I'll talk in quite a bit of detail about them. I would like to call out that these are all, this entire activity is funded by NCATS. That's one of the NIH institutes. And it's run through the National Center for Data to Health that I already mentioned two of the PIs, Chris Shute and Melissa Handel. So N3C was really set up to try to be a better place to both develop, release, and validate various kinds of algorithms to understand what's really going on with COVID-19. Of course, support drug discovery and pharmacogenomics. A hope was it would be useful for multimodal analytics. So not just EHR data, but also imaging data, genomics, that digital health data that I mentioned. And that there would be an understanding of which interventions really reduce disease severity. As we found, right now, the best intervention that we found so far is vaccination. And that has had an incredible impact on disease severity, even for breakthrough patients. And some best practices for making sure that we can allocate resources and make sure that, in fact, we can support reproducibility. So I'm not going to dwell on all these questions. I will come back to one particular question, though, and that's looking at cancer patients and is cancer and cancer therapies, do they cause increased risk for cancer patients? But some of the questions that N3C would like to be able to support is things like social determinants of health. Which ones are risk factors? Which ones increase severity? Which ones may impact mortality? And do therapies work better than, some therapies work better than others? And an interesting part of that is, do some therapies work better either in geographic regions or in certain demographies, in different settings? And can we compare local clinical observations across the whole country? And then moving more into the prediction realm, can we start to predict who will have a severe outcome if they get COVID? And can we then help prioritize, for instance, who gets vaccines, who gets more immediate care when they would come up with, when they would get a COVID-19 infection? Some of the things that we're still trying to do are things like, can we predict acute kidney injury in COVID-19 patients? And that's, I won't talk more about that, but that's been a big issue in many COVID-19 patients and who might need a ventilator. And again, as COVID-19's evolved and our ability to care for patients has evolved, some of these issues have come in and out of focus. So ventilators, for instance, are much less a problem today than they were a year ago, because we're doing a much better job of managing COVID-19 infections and even severe infections. So I'm gonna kind of skip over this because I've mentioned pieces of it, but the middle bullet I want to talk a bit about, and that's that the N3C cohort itself is all available in OMOP. So that is one of the common data models. It was developed by the Odyssey Consortium. And it is the way that, in fact, all the data from the different projects I'm going to talk about, sorry, the different organizations I'm going to talk about are brought in and then made consistent so that they can be consistently analyzed. And then just thinking about benefits to participate in N3C for anyone who wants to do that. It's great for pilot data for grant proposals. It really is that big data resource. And it's a way to also do team science. There's lots of folks involved in N3C, and you can work hand in hand with many different kinds of individuals with different training. And it's a great opportunity to use machine learning analytics and things like natural language processing, and of course, access to lots of tools and software, and additional data sets that have been brought into N3C. So I'm not going to talk a lot about this, but it's worth spending a little bit of time on this, that the cohort of patients that are COVID positive in N3C, it is based on them having in their clinical record a clear COVID-19 PCR test, and then a positive result. And at the same time, the entire cohort has demographically matched samples where 2 additional patients from each of the organizations that's contributing patients, they have very similar patients from a comorbidity, an age, and a demographic standpoint, a socioeconomic standpoint, that have also had COVID tests but have been negative, are pulled in as matching controls. So the other piece that's really important is the data that goes back to January 1st, 2018, and is pulled forward. And the reason for that is we have then a few years of longitudinal data from the EHR and every one of the patients that gets matched. And that obviously depends on the care setting and exactly who they are, whether they've been seen frequently at a given care site that's taking care of the COVID patients or not. And as you'll see, in fact, there's deep data on most of the patients. So that just gives you a sense of who is eligible to come into N3C. This is the timeline up until March. Apologize for not having updated graphic that takes us through to today. But you can see in March, N3C hit 1 million COVID positive patients. And this next slide, you'll see that as of yesterday, there were 6.3 million total patients. Remember that for roughly every patient with COVID positive, there are 2 that were COVID negative. So there's a little bit more than 2 million COVID positive cases today. So just 2 months later, after we hit our first million, we've now hit 2 million. So that says that we're still having many COVID cases in all of our locations. And then 2, we're also still expanding the number of organizations that are submitting data. So both of those are responsible for the uptake. There's almost 7 billion rows of data. And just to give you a sense of where all those data are, There's almost 600 million clinical observations on those 6 million patients. So that's roughly 100 observations per patient. There's 3 billion lab results, right? So that's almost 1,000 lab results per patient. The number of medication records, and remember, this is just over a 3-year period, is roughly 1 billion. So again, that's almost now 200 medications for all those patients. Medication records, not necessarily individual medications, and then 300 million procedures, and it covers about 300 million individual visits. So, and I'll talk a little bit about what a visit is, because I think that's important, and also shows a limitation of EHR data. And here you can see the 55 sites that have released data into N3C and their locations. Ocean is a network that's located across the whole country. They've been submitting data from almost 100, well, over 130 sites, and that's also part of N3C. So I won't have the chance to talk about this, but there's also 31 different clinical domain teams that are working on different clinical areas using N3C to really understand things like acute kidney injury, and ventilators, and all the different clinical questions that people have. So there, as of yesterday, there were 88 data transfer signatories. So these are all the organizations that have agreed to submit data into N3C. And it's worth noting that 55 of those have actually had their data now in N3C. So there's still a number of sites that have committed that are in the process of having their data flow into N3C. And I'll say that for Duke, we did all of our legal agreements and regulatory agreements back in June, and it took roughly 2 months for us to be able to give our first data set and to go through the entire harmonization pathway and get into N3C. So there is a lag from when people agree until data flows in. I do want to talk a little bit about the data stats for the enclave. So you can see that looking at men and women, roughly the same distribution by age. Notice that at 90, there's a cutoff, and that's because due to HIPAA regulations, everyone who's over 90 just gets coded as 90. And you can see that again, there are a number of pediatric cases. So the COVID-19 does hit the under 18 age group, not with the same frequency, but it is not an insignificant disease for children either. Just to see those in numbers, you can see them laid out here, the COVID cases in the left data column, and then the breakdown by race. Something that's, I think, really worth talking about is unlike what we would have seen 10 years ago, is we actually have pretty good capture now of race and ethnicity in EHRs. I will say that a few institutions didn't do as good a job at this. So the numbers you see are the unknowns are driven by a few institutions, not the many. These are, I should point out that these are actually available, and I think I put the URL somewhere. Maybe I didn't, I'm sorry. But if you Google N3C dashboard, these are public dashboards, and you can actually see all these data publicly available, see the latest. And this is the comorbidity distribution of COVID across the EHR. What I think is really remarkable is the top two, diabetes and renal disease. So looking across the institutions, and this column's not here, but roughly 10% of all patients that we were submitted to N3C have diabetes. About 2.5% have renal disease. But when you look at the severe cases, they are highly enriched for diabetics and for patients with renal disease. So it's very clear those are really very important risk factors for patients. And you'll see an enrichment in cancer, but when we dig into those numbers, you'll see that they don't quite, it's not quite the same as we see in diabetes and renal disease. And we'll get into that a little bit more. I also want to point out, even for diabetes and for renal disease, the characteristics of some of the patients that experience mortality is what you'd expect for high mortality patients. So again, that's an important caveat to all this. Is it's really important to understand the characteristics of the cohort when you're evaluating whether or not something is important or not. So this is just a slide from the paper that went in back in January, which seems like such a long time ago now from a COVID standpoint, but it was really the first characterization. And you'll see that a lot of the numbers that are now publicly available, we really developed the algorithms and the methodology for analyzing the N3C cohort as part of this paper. And now it's all public. So encourage you to go read that paper. If you're interested, if you want to Google it, it's the National COVID Cohort Collaborative Clinical Characterization and Early Severity Prediction. And at least as far as I know, it's still a medRxiv preprint. It has not managed to go through every step of the review process yet. Again, that's important that N3C is actually going much faster than the current publication methods. And we've seen that with many COVID papers, that in fact the preprint archives have been incredibly important. So I've gone through high level purpose for N3C, who the contributing sites are, what the inclusion criteria is to have a patient represented, either as a control or as a COVID positive patient. And you got to see the dashboard. So I'm going to talk a little bit about data ingestion. I probably won't talk about data governance and security. I'm too long-winded for that. But then I do want to go through the discussion of cancer and COVID-19. So I already mentioned the 4 common data models that are in use, and different organizations use different models internally to be able to organize their EHR data and make it available for different kinds of research purposes. So the good part about N3C is we can take data from any of those models and import it. They all, though, do have differing characteristics. So I think it's important as we talk about harmonization to realize that piles of parts, like shown in the upper left, is not as valuable as having a catalog of parts, and that having that classified data is incredibly important. And so N3C spent a lot of time developing that classification. And there's really 2 pieces of it, and I'm not going to go into the details of how to do it. That'd be a whole talk all by itself. But one is syntactic interoperability. So is the structure appropriate? You can really think of that, the classic syntactic interoperability is the different kinds of plugs that go into the wall for electricity. And different countries have different styles of plugs. Then the semantics is the meaning of what you're exchanging the same. And to take that plug analogy a little further, some countries use 220, some use 230, some use 50 hertz, some use 60 hertz. And you have to have a device that's capable of handling those different voltages and those different frequencies to give you power when you plug in different countries. So you have to have both the syntactic interoperability, and then you need to have semantic interoperability. And both of those are necessary to be able to do something like N3C. So just to show you in a graphic form, we have the ability to bring in the OMOP data, the ACT data, the Trinetics data, the CoreNet data, go through what we call an ingest server, parse it all out, start to move it into OMOP, right? Prep it, move it into OMOP. We do a lot of things with mapping, making sure that the value sets are all appropriate. Build the primary keys so that we know which projects and which organizations have submitted which things, but we don't release that in the aggregate N3C data. So every organization is actually blinded to which organization submitted the data. And we move from this idea of what initially starts out as manual curation into having this big data industrial scale ability to pull data in. So, excuse me, what I didn't mention is that for instance, for Duke, that first process was pretty long, bringing data into N3C. But once we've gone through it a few times, it was all completely automated, and it now only takes a few hours to bring in all of our data, put it in, and release it into N3C. So that's that moving from manual curation through to industrial scale harmonization. And that's incredibly important. I'm not going to dwell on this, but just to say that the different common data models, they actually have different purposes behind them. And so they don't actually handle data quite the same way. And that's both good and that's bad. It's good because they're good at certain things. It's bad because when we want to bring them all into OMOP, they don't all match. So just to realize that there can be differences in purpose. The other piece, and I'm not going to really discuss this, is that the enclave itself is incredibly secure. It's managed by NCATS, managed by NIH. It meets all the federal security standards, and you have to have you need to be registered and onboarded to be able to use the resource. So even though now a lot of the data, a lot of the aggregate data is made publicly available, if you want to get to patient level data, you do need to go through process. Yeah, I'm probably going to skip over these points, but I do want to make a really quick point about federated versus centralized, because I've been involved in both kinds of projects, both building federated resources and then building centralized ones. For N3C, it's been very important that it's centralized. And one of the reasons for that is, in fact, that those 4 different common data models all make different assumptions about underlying data in the way that even things like the individual patients, their encounters are handled. Excuse me. So I'll show an example of how that works in a second. So N3C is centralized. There's absolutely nothing wrong with federated networks. But it does different things. They do different things. So for instance, now we can take an example of 3 sites that say that they have, they have very different numbers for how they think about their patients and their visits. So for instance, here we can see that this 3rd site, basically every time a patient walks in the door, or every day that patient is in an inpatient setting, they treat it as a separate visit. At the other sites, they're not treating it that way. Right? They're they're saying when the patient comes in the door until the time they leave, that's a single visit. So by having all the data in one place, we can actually process it, we can think about it, and then we can harmonize the data from that third site so it looks like the data from the others. So that's one example of something you can do when the data is centralized that's very hard to do when it's been federal or when it's federated. Another thing that's been incredibly important for N3C is thinking about unit harmonization. So here's an example of body weight, and each of these violin plots are coming from different sites. These are actually real violin plots. And you can see that not surprisingly, sites that say they're in grams, the values are higher than the sites that say they're in kilograms versus ounces versus pounds. And what may not be apparent here, but in fact, this one site wasn't specifying which one it was. And depending on the clinic that the patient was seen in, they were entered in ounces or in pounds. So we're getting both. So that represents a data harmonization problem of something really simple, right? And there was actually a pretty sophisticated algorithm that had to be put in place to be able to deal with this. And once that's been put in place, now you see that in fact all the violin plots look the same and they're on the same axes, and it's clear exactly which unit they've been converted into. So that's just a single example of hundreds of different measurements that have been put through this process. And I think what's also important is there are ways to both visualize these kinds of data and identify the ones that need this kind of algorithmic help. Again, I'm not going to go into this in a lot of detail, but the short answer is that some of the work in these little black boxes are saying that we actually can't do that automated conversion. And it's a data quality issue, and we have to go back to the sites and say, please clean this up. So the other thing I want to talk about very briefly with N3C is phenotypes, and thinking about how do you harmonize phenotypes. So, and by phenotype, it's really now, if you will, a little higher description of something that's experienced by a patient. And some of the magic, frankly, in doing this, and a lot of this was done with the human phenotype ontology and with the Monarch initiative, is coming up with human readable, human understandable definitions, because not everyone is a clinician that uses these resources. So again, going through and making sure we have pretty clear definitions for all the terms. And as you can see on the left-hand side, relationships between all the different phenotypes and the phenotype group they're being mapped to. And what is important is that phenotypes can map to multiple groups. Right? So again, this is another way of being able to help people when they want to analyze data that's in N3C, to identify where the data they're interested in resides in the EHR. So something that we will be doing more of is moving into FHIR. So that's FHIR is a HL7 standard for making EHR data more accessible. And that's it's really a syntactic change, making sure everyone uses similar, if not identical, common vocabularies, and leveraging Ascudi. So that's the US Common Data Initiative, and that's again incredibly useful for the semantic modeling. So as more and more groups start to leverage Ascudi, some of what N3C has done will become easier and easier to do again. I'm not going to talk about governance. I'm just going to skip over all these slides. See, sorry, don't close your eyes if you get easily dizzy. I'm sorry about that. Didn't mean to do that. I did want to talk very briefly, though, about team science, and I think it's relevant for the oncology discussion. And again, you know, NCATs. So for those of you who are familiar with NCATs, they've been one of the earliest and biggest supporters of team science at NIH, and it's really about supporting all the different kinds of expertise necessary to do research in today's world. And you know, here are a few of the example key functions and the potential domain teams that need to work together to really make things like N3C work. So I'm not going to belabor that more than what I just said. Now I'm going to turn to the oncology research paper. And this is a presentation. I'm going to give you a presentation that Noah Sarah Feldman gave. Again, she's at UAB, University of Alabama at Birmingham, and a lot of different people, and including Umit Tabaloglu at Wake Forest. And they did a really nice analysis of N3C to understand if cancer patients were really at greater risk for COVID-19, and in particular, more severe outcomes. It's just to show you where the members of the team, not just I should have also mentioned, recognized Benjamin Bates at Rutgers. The three of them were really foundational in making the oncology domain team work. And you can see domain team members are from across the country. And back to that team science idea, is there are a lot of different pieces of expertise that need to be in place to make an analysis like this work, right? So there need to be people who understand the clinical data, understand how to build the necessary infrastructure just to make the queries and the analysis happen. Making sure that we're not making silly statistical errors, and biostatisticians are critical for that. Understanding from an epidemiological standpoint, well, what's going on in the population? And then using actually the N3C resource. And you can see that, excuse me, that actually requires a pretty big team, because doing things in a secure cloud is not trivial. So that's an example again of it. The team science is necessary to make something like this work. So I'm pleased to say that the team published their work at the ASCO meeting. So those of you know the American Society for Clinical Oncology. The meeting was at the beginning of June, and the publication came out simultaneous with the presentation. So this is what they did. They needed to define all of the concept sets that would go into defining a cancer patient, because that was something that, at least inside N3C, had not yet been done. So they followed a lot of both existing work that SNOMED teams, that UMLS had done, and the ICD teams had done, and make sure that they could distinguish between the various cancers. And interestingly, the overarching malignant neoplasm concept code still included 12 terms that actually weren't malignant. So those had to be manually pulled out and manually excluded from the analysis. Or at least manually annotated. It was all done automatically, but manually annotated. And then from a disease standpoint, defining the anatomical sites, trying to find primary mentions of diagnoses. So for those of you who have looked in EHRs, sometimes it's hard to figure out what the primary analysis or the primary tumor was. And then one of the important parts is come up with the diagnosis code for the critical visit. And the critical visit idea is somewhat specific to this kind of research, but it's creating an index date and then making sure everything is analyzed with respect to that index date, and then have a final primary diagnosis based on all that analysis. So that's looking at now the cancer cohort. Once that's been created, then just really it's applying that to all the N3C patients. At the time that was, and this was now 2 months ago, there were about 4.4 million cancer patients. And that created then the cancer cohort. There were about, at that time, about 400,000 patients who had cancer, who were part of N3C. All pediatric cancer cases were excluded, which left just under 400,000 patients. Then it was partitioned into COVID negative and COVID positive. And the positives, again, back to that index date, they were selected down to patients that were given a COVID positive diagnosis within 21 days prior to that index date and 5 days afterward. And that left them with a cohort of 38,000 cancer cases. And the analysis I'm going to show was really looking at that 38,000, those 38,000 cases. So from just to give you a sense of the characteristics of those 38,000, you can see they looked about like what you'd expect. Very few patients under 30 because it's rare for people in that age group to have cancer. Almost exactly split based on male and female. Geographical location is, I'll say, an artifact of where the sites are that are providing data to N3C. So this actually corresponds roughly to the size of the patient populations that are being sent to N3C from each of these geographic areas. And then the Hispanic, non-Hispanic, and then unknown race of the race and ethnicity of the individuals that had cancer. So about what we'd expect. So looking at the Charleston comorbidity index, 0 means they're very high. They're very healthy. And then as you see, increasing comorbidity index, that means that they're doing poorer and poorer. Typically for clinical trials, for instance, most clinical trials cut off at Charleston index of 2. So the patients that are above 2 aren't eligible for clinical trials. And then just to see how many were smokers, most were non-smokers. Then diving into where the different what cancers are represented from the solid tumors standpoint. Here's the percentages. So you can see that it's split between different skin cancers, breast cancer, prostate cancer, hematologic malignancies, gastrointestinal cancers, and multi-site. That's not all of them, but that's the vast majority of the cancers represented. Again, a pretty, if you will, a good sampling of cancers by common, of all the common cancers. And then looking at the treatments that those 38,000 patients had experienced. And the treatments, again, they're in that index period. So whether they were getting systemic antibiotics, roughly 15% of them had systemic steroids, AZ, remdesivir, dexamethasone, or hydroxychloroquine. And again, you can see that there were very few that were given chloroquine. And then looking at the death and invasive ventilation numbers, you can see that COVID positive patients are roughly at a 2% increased risk of death. and at roughly a 3% increased risk of invasive ventilation. So those are very similar to the risks of patients that are hospitalized, period, with COVID-19. So not the 14% and 8%, but rather the differential. So that says almost immediately that that getting COVID while you are a cancer patient probably doesn't impact overall your risk. But this is of course now for 19,000 of those patients. So these are the ones that experienced, of those 38,000, either death or invasive ventilation. Looking at it a different way, just looking at the survival curves, you can see that there's roughly a hazard ratio of 1.2 if you are COVID positive of increased mortality. Again, that's roughly in keeping with the hazard ratio of getting COVID-19. And then looking at this now by individual cancers. And this is, I'm not gonna say that this is misleading, but what they didn't do is show the survival curves for the patients that weren't COVID positive, so the COVID negative patients. And again, looking at this from a hazard ratio standpoint, you can see there are relatively few patients that are at highly increased risk. Again, over 65, they are notably at risk if they're cancer positive and they're COVID positive. But that's not, again, I'm going to say that's not completely unexpected, because everyone over 65 is at pretty substantially increased risk. This is a little bit interesting because it shows that if you live in the West, at least according to the patients that came in, you're at increased risk of dying if you are if you have a cancer diagnosis and you're COVID positive. And then again, the Charlson comorbidity index numbers, as you're having more and more comorbidities, not unexpectedly, your hazard ratio goes way up. So, and now looking at this from a cancer treatment type, if you're on at the time, again, during the index period, if you're getting cytotoxic therapy, that is a increased risk. If you're on a targeted therapy, that's an increased risk. Immunotherapy seems to make absolutely no difference. Interestingly, though, endocrine therapies appeared to reduce overall risk. So that actually, all of these things did correlate and actually were validating of different publications that were made. So again, it looks like from a treatment standpoint, these individual therapies really didn't seem to make much difference. You'll see that it looks like, for instance, systemic antibiotics looks like an increased risk. But again, keep in mind that some of these increased risks can also be because the patients that were given these were given them because they were already more fragile. So again, that's an important caveat to all of these kinds of analyses, is you have to pay attention to the cohort you're looking at and their overall frailty. So just, and this is from them, their limitations, the real world data challenges. So it's clear that probably in their cancer, that not everyone who is in the at the time they did this 4.4 million patients was in fact cancer negative. So that's the data missing this issue. But in fact, they weren't really using those data anyway, so that's not really much of a limitation. Limited capture recent cancer therapy, I'm not sure how big a limitation that actually is. They couldn't quantify it. I think the misclassification piece was they did actually a remarkably good job of that. And I think they actually did a pretty good. They paid a lot of attention to making sure that these things were well controlled. So as I mentioned at the very beginning, the conclusions were that one, N3C is a unique resource. It does have a lot of patients in it, and that it's the largest COVID-19 and cancer cohort. And that consistent with previous literature, older age, particularly male, with increasing comorbidities and with hematologic malignancies were associated with higher mortality. And the N3C data set seemed to indicate that immuno, and actually this should be endocrine, that I hadn't caught that in the slides, but immuno or endocrine therapies were not at higher risk. So that was the summary of a lot of work that the oncology data team did. Their thanks to everyone. And you can see again from a team science standpoint, just how many people were involved in making all this happen. And these are folks. They interestingly even thanked me for being involved. So they were very thorough. I'm gonna stop talking here in a minute and answer all your questions. But just in case you're interested in using N3C, and all these slides will be sent to you, and all the links are live in the slides. But it's very easy to register. You just Google N3C registration, and it'll take you right to the registration page. There's lots of resources for learning how to use it, and joining the domain team is a great way to get coupled to other people who already know how to use N3C. So I'm gonna stop there, and here are all the other resources for getting involved with N3C. And I will stop sharing and happily take questions.
Thank you. I have actually a question that's more on the logistics. So you said the project really spanned 3 years. So how long then was spent initially, like on planning how the data was going to be collected and curated in order to make it interoperable?
Okay, so sorry, I misspoke. The EHR data coming from each of the sites, each of the organizations that's participating in N3C, they go back, they actually mine retrospectively data on all the COVID tested patients back to January 1st of 2018. N3C didn't no one thought about doing something like N3C until April of 2020. So that's when the planning started. It went from planning phase to reality in about a month and a half. So that was that's certainly the most rapid project I've ever seen. So I don't know if that gets if that's the answer to your question or not.
Yes, thank you. We do have, we unmuted.
Yes, that's great presentation. Thank you very much. So the data on cancer patients definitely very interesting. And when they are mining the data, some of the cancer patients, of course, they are survivors, and some of the cancer patients, they are under treatment, as you presented, immunotherapy and then chemotherapy. Is also there like radiation treatment or radiation therapy, these kind of also groups also included in N3C, and also like alcohol cancer patients, those who are also addicted to alcohol, and it also weakened their immune system, and it also really helped, was the also cause of the mortality. So is this kind of also added into the N3C or planning to add into N3C?
So let me address the first point first. So cancer survivors were not included in the analysis. So that was and I'm sorry, I really jumped through the importance of the index date. So the index date said that they had to be getting an active therapy for cancer within that window. And it was only a 30-day window. So they probably left out a lot of patients that, for instance, are in between cycles of chemotherapy, where they've gotten a set of chemo, and now they're going to go 2 months later and get radiation. If that window had been right when they got their COVID-19 diagnosis, they would not have been counted as cancer patients for the purpose of the analysis. So the analysis really is only of active cancer patients that are getting active therapy. To your next point about alcohol use, the problem with that in EHRs is it's not well captured. I think we've all been to our primary care doctor for our annual exam, and they ask us how many drinks of alcohol we've had in a week. And those of us that perhaps drink more than we should, we probably underreport. There's no way to, that's just not, unfortunately, that's not an accurate part of the EHR. Smoking history is because of the repetition is actually a pretty good place. So smoking history, I feel is a little more consistently asked. Even there, though, I'd really like everyone to ask pack years history. That turns out to be the real risk. If you have 10 pack years smoking history or 40, those are important numbers. But just knowing how frequently or if you were a smoker or not isn't quite as valuable. So even with smoking, there's some limitations. Those are great questions, but unfortunately, those are limitations of EHRs, right? We don't capture a lot of what I would call the more social factors and behavioral factors, as well as we probably should. And in part, so this is now the philosophy of your question. In part, the reason we don't ask those questions is we don't actually know how to intervene. So that's part of the problem for most of our care organizations, is unless there's something they can do, they typically don't ask a question.
Thank you.
Absolutely, that was a great question. Thank you.
And Professor Minjiang asked, said, great talk. I'm wondering if there's any molecular data available.
So by molecular data, do you mean sequencing data, or what's molecular data?
Yeah, like sequencing data, say to look at the risk prediction or response to certain treatment, comorbidity, basically is a kind of a genetic basis for all this.
Yeah, so there actually is a sequencing technologies domain group. The problem is it's twofold. One is there's not a lot of, outside of cancer, there's not a lot of sequencing data on most patients. And then two, when it comes to sequencing COVID itself, so looking at the viral sequence, the US is so far behind a number of other countries that it's actually a little sad. So for COVID-19 in particular, most of the characterization's just been PCR, and even for understanding the variants, it's all been PCR based assays. It hasn't been really sequencing the genomes, the viral genomes, or the genomes of the patients. So there's been discussions of trying to set up more sequencing data. N3C is going to be working with NHLBI, so another NIH institute, to be able to try to walk between all the sequencing data that NHLBI has been doing in primarily for cardiovascular risk, and then try to do crosswalks to see if there are variants present in some of those cohorts that would explain either really good outcomes in COVID-19 or really bad outcomes. But that hasn't happened yet.
But then the current collection is mainly just data or also some samples, so like biological samples from these patients?
So there's N3C currently does not have an independent biobank. And actually, I would be surprised if N3C would be the one to build that biobank. But I guess to answer your question, there are now several projects. One's the one I mentioned, trying to cross top med data from NHLBI with N3C. So that's where those all that sequencing data is, and actually a lot more characterization of those patients as well, or those individuals. But it's also crossing with imaging data that's in MEDRIC, which is at the University of Chicago, and imaging data in TCIA, Cancer Imaging Atlas, and then trying to crosswalk between those resources so you may be able to get imaging data on specific patients, and then see and be able to then enrich it with all the clinical data in N3C. So N3C really is focused on what comes out of the EHR. And not so focused, but would like to have access to all the other kinds of data.
Thank you.
And we have a couple questions too in the chat. Go ahead.
Yeah, so Joanne, that's a great question that is part of the survival difference, because the hormonal therapies could be could that increased survival be because the cancers themselves have high survival? I don't think so, because we're comparing with survival rates for COVID-19 as well. But that's a great question, and I wasn't involved in the actual analysis, so you're making me wonder how they did those hazard ratios. Because that's really what you're getting at, are the hazard ratios relative to, and I think this is true, relative to patients that didn't get COVID-19 and is somehow, are those cancers somehow selecting for a healthier population? That's a great question. Again, it goes at that selection, potential selection bias. Savannah, the future applications of the study. So do you mean the oncology study, I presume? So one is, it's interesting because as soon as they published it, there's almost twice as many patients now available as when they started the analysis. So I'd love to see them just replicate it now that there's so many more patients in N3C. But I think that they have a number of other clinical questions that they're asking, and some of them are more detailed around the therapies. So I think they're going to do some more detailed understanding of the risk of some of the therapies that are given to the patients, and see if they can tease apart differences. And that'll work better as they have more and more patients. And Joanne, I think you have the ability to speak now.
Oh, okay, thank you. I was just going to say, even within breast cancer, the ER positive tumors are the ones that are going to be treated with hormones, and those have the best survival of all breast cancers. So it's kind of a tricky thing to pull apart. That's all I was going to say.
Yeah, no, you're making a great point. And I again, we'd have to go. Maybe you should join the oncology domain team and help them ask that question in other ways.
I would
be able to
happy
tease that apart. I'm sure they would love that.
Can I send you an email and find out who to contact?
Absolutely.
Okay, great. Thanks.
And then we have another comment/question. So thank you so much for this great presentation. Have a question about geographic locations. 4 to 5 geographic locations were the smallest geographic unit that is available from the data. And are there more fine-grained data available?
So actually, the home zip code of all the participants is in the enclave. That's in the most protected part of the enclave, and we can't expose those in a presentation. Can't expose them directly in an analysis. So it actually is there, and what I didn't get a chance to talk about is some of the social determinants of health research that's going on in N3C. They make a lot of use of the zip codes and when it's available, 9-digit zip codes, to be able to really hone in on things like the American Community Survey data for that particular location, and be able to understand the context of those patients, those COVID-19 patients, to understand if they're at increased risk, decreased risk. Are there protective factors? Are there risk factors around them that we can start to tease apart? So that's a great question. That's one of the most protected parts of N3C. I should say that even the most protected parts, though, don't have patient names, and but it is zip code and potentially even 9-digit zip, and those are very identifying. So you have to get you have to have an IRB protocol and have that go through the full process to gain access to those data. And you have to have very clear scientific question that needs access to those, like the social determinants of health kinds of questions.
And then the last part of their question is also on whether there is information on stages of cancer.
Yeah, so I kind of glossed over that in there was part of the flow sheet where they were trying to get at staging. If you've dealt with EHR data, staging is poorly captured in the EHR. In some cancers, it's captured pretty well, but generally it's not well captured. I'm looking forward to the day where EHRs actually use the new PRISM model that Deb Schrag at the Dana-Farber developed. And we actually have that coded in the EHR, because I think that would really allow us to do a lot better job of staging, grading, and understanding where in their trajectory. So that's the other part of this, is just because they had a therapy, we don't really know where they are in their therapeutic journey. We just have this little snapshot. So EHRs are imperfect tools for helping us really understand all that, even at scale. But the good part is when you have enough patients, like N3C has, you can ask questions and still get, I think, meaningful results. And let's see, it seems like the questions have died down. Any last questions for me?
Yeah, I don't see any other questions coming in, just some thanks.
All right, well, you're all very welcome. Hopefully you found that useful. Please join N3C. I think it's a great resource. And for those of you really interested in big data, it's a great big data resource. So what I didn't really, I didn't say too much about this, but it's actually everything in N3C is cloud-based. So it's a cloud computing infrastructure. It is FedRAMPed. So back to the security pieces, that's one of the things that NIH, actually the federal government, requires. If you want to keep federal data in a cloud computing environment, it has to be FedRAMP. It's a pretty high bar. So I feel pretty good that in fact, patients' privacy is well protected. And of course, because it's a cloud environment, if you want to do some pretty intense computation, it's available. And I guess I didn't say this up until now, but NCATS foots the bill for that. So if you run a really complicated computation that takes a lot of machine hours to do, NCATS is covering all that for you. So you get that's a tremendous resource.
Wow. So basically we get free computation.
That's right. For now, it's free computation. And talking with the folks at NCATS, they're paying more and more money every month for people to do computation in it. Part of that's because, of course, as the data sets gets bigger and bigger, it takes more computation to ask the same question because you're going through bigger data set. But it's also that there's now, and I didn't focus on this, I think there's more than 1,500 people that go in a given week into N3C and do something. So the number of researchers in there, it continues to go up. And I see a question from Young Xiao. So disparities in healthcare utilization, I don't know if anybody's asked that question. The closest, the folks that are doing the social determinants of health are getting at that question from a little different angle. They're trying to understand if there are overrepresentation of particular groups or underrepresentation. And I don't think, so part of the issue is because this is not a truly representative sample of the US, it's hard to ask those questions because you don't know if someone hasn't been seen. Let's say, here Duke, we provide our data to N3C, as does UNC, which down the road. But the other healthcare providers that are providing healthcare in our area are not providing data. So if you saw a disparity, is it because they're actually going to a different care organization, or is it truly a disparity? So I think that's a hard question to ask, and that's really something that, again, looking at it from a social determinants of health standpoint is probably the best way to get at those questions. Again, I encourage you to get involved in N3C. Join the SDOH working group.
Okay.
Right.
Yeah, thank you so much for another wonderful talk again for our workshop, and yeah, we really appreciate your continued support of our special lecture series. So with that, thank you everybody for joining us today.
Thank you, man. Thank you, Nadia. And thanks, John. Appreciate you making sure that I'm not disconnected. Take care, everyone.
Thank you. Bye.
Thank you.
Bye.